This training course is designed with a singular focus: to empower clinical investigators with a thorough understanding of trial design, regulatory requirements, and innovative strategies that bolster trial efficiency. Such knowledge is not merely beneficial, it is vital for elevating the quality of medical products that can ultimately transform patient care. In outlining the course structure, we delve into a series of well-crafted sessions that concentrate on both clinical trials and non-clinical data. This approach aims to instill confidence and competence in investigators, enabling them to conduct clinical research with effectiveness and conviction. We encourage active engagement through questions, as interaction is the cornerstone of collaborative learning. Dr. Sachs sheds light on the historical context of FDA regulations, reflecting on past public health failures that served as catalysts for implementing strict drug safety laws. Understanding this rich history is crucial, for it lays the foundation for appreciating the regulatory practices that govern today's healthcare landscape. The FDA's remarkable workforce comprises over 18,000 dedicated individuals, many of whom are scientists committed to safeguarding the safety of medical products. Their relentless efforts oversee a vast spectrum of food and drugs worth over $3.6 trillion, underscoring the gravity and scope of their mission. The legal framework that guides the FDA's pivotal work includes the Federal Food, Drug, and Cosmetic Act alongside the Public Health Service Act. These statutes define the authority for clinical investigations, while the corresponding regulations detail the practical implementation of these laws, ensuring a thorough compliance landscape. Central to initiating clinical studies, investigational new drug applications, INDs, empower sponsors to transport products across state lines for research purposes. This mechanism serves to safeguard the rights and safety of participants involved in clinical trials, affirming our commitment to ethical research practices. Clinical trials are tasked with delivering comprehensive data on drug safety and efficacy. They employ rigorous statistical analyses to validate findings, making this information indispensable for regulatory decision-making, which ultimately affects patient access to new treatments. The regulatory frameworks, such as INDs and NDAs, serve as robust mechanisms for overseeing drug development and securing market approvals. They ensure that investigational drugs adhere to the highest standards of safety and effectiveness before they reach the patients who need them. In the realm of clinical studies, the inclusion of control groups stands as a fundamental component, essential for differentiating treatment effects from other influencing variables. Thoughtfully designed investigations minimize biases, thereby yielding reliable and actionable results. The significance of control groups cannot be overstated. Their role in minimizing bias contributes to the robustness of study outcomes. It is noteworthy that different control methodologies can lead to divergent conclusions regarding treatment efficacy, emphasizing the need for careful study design. Placebo-controlled trials provide a unique vantage point, enabling researchers to discern between the effects of the drug and the natural progression of disease. A profound understanding of the role of placebos is essential, as it clarifies the actual benefits that new treatments can offer. Employing enrichment strategies within study populations aims to enhance the likelihood of detecting drug effects. By reducing variability or concentrating on patients predisposed to respond positively, these strategies significantly elevate the potential for meaningful and actionable outcomes. Prognostic enrichment is a strategy that focuses on selecting high-risk patients for hospitalization. This approach not only improves trial outcomes but also enhances the overall relevance of the study, taking into account the patient's medical history and characteristics. On the other hand, predictive enrichment hones in on patients likely to respond to a treatment based on specific biomarkers. This targeted methodology ensures that drug efficacy is assessed within the most appropriate patient population, leading to more reliable conclusions. Randomization is a crucial aspect of clinical trials, minimizing bias while ensuring comparability across groups, an essential aspect for achieving valid outcomes. This process plays a pivotal role in eliminating confounding factors that could distort the results. Blinded assessments serve to further mitigate bias by keeping evaluators unaware of treatment assignments. This method aims to enhance the objectivity and integrity of the results garnered from the study, thereby reinforcing the reliability of findings. The judicious selection of appropriate endpoints is imperative for aligning with the study's objectives. These endpoints must hold significance for the target population, as they provide crucial evidence that informs the overall goals of the research. Intercurrent events pose a substantial risk to study outcomes, necessitating their careful consideration during the design phase. Identifying such events enables researchers to accurately interpret trial results, making this foresight vital in the research process. Crossover designs present an innovative approach, allowing participants to receive multiple treatments, which can yield more reliable results by minimizing individual differences. This type of design enhances the overall assessment of outcomes, fostering a comprehensive understanding of treatment effects. However, Carryover effects can introduce complexities in crossover studies, as prior treatments may inadvertently influence responses in subsequent phases. Researchers must strategically plan washout periods to mitigate these effects and preserve the integrity of the findings. The prompt observation of endpoints is of paramount importance, particularly in chronic diseases where timely data collection is critical for achieving accurate outcomes. This need for urgency underscores the importance of strategic planning in study design. A robust grasp of statistical concepts is indispensable for effective study design and analysis. Addressing issues related to bias, variability, and multiplicity during trials presents challenges that researchers must navigate with precision and knowledge. Minimizing bias and variability stands as a cornerstone for conducting ethical and feasible studies. Striking a balance between these often conflicting goals requires vigilant oversight and strategic planning to ensure the integrity of the research. On the other hand, enrichment strategies can amplify the statistical power of trials, though they may inadvertently hinder generalizability. Crafting results that resonate across diverse patient populations demands careful consideration of study design elements. A larger sample size is synonymous with improved estimates of the average, as it mitigates variability among samples. This principle is vividly illustrated when comparing the outcomes of samples consisting of 5 individuals versus 50. The principle of standard error further illustrates this concept. As sample sizes increase, so does the reliability of results. Mathematically, this relationship is represented by dividing the standard deviation by the square root of the sample size, emphasizing the importance of scale. The z-statistic serves as an indicator of the significance of an effect estimate. Notably, larger sample sizes yield smaller p-values, which suggest more substantial results. This serves as a testament to the critical role sample size plays in enhancing statistical power. Confounding arises when baseline differences exist between treatment groups, leading to skewed results. Thus, randomization becomes an essential strategy to effectively mitigate this form of bias, ensuring fair and reliable comparisons. Measurement bias can emerge from issues surrounding endpoint ascertainment and is often addressed through methodologies such as blinding and reliable measuring techniques. Adopting robust methods is key to preserving the integrity of study outcomes. Selection bias poses a threat to the generalizability of study findings, especially when the study population fails to be representative. This distortion can impact the validity of the conclusions drawn, emphasizing the need for careful participant selection. Patients may discontinue treatment for various reasons, including adverse events or perceived ineffectiveness, which can compromise data integrity. Recognizing these factors is essential for conducting accurate analysis and drawing valid conclusions. Different types of adherence challenges, such as treatment discontinuation versus missed study visits, should be categorized appropriately for effective resolution. This distinction is vital for ensuring unbiased results that reflect true treatment effects. Intent-to-treat analysis emphasizes the inclusion of all patients, regardless of adherence levels, thus preserving randomization. This methodology proves effective in evaluating the impacts of the therapy assigned while minimizing potential biases. The principle of randomization holds immense significance in clinical trials, as it is instrumental in reducing baseline biases. It ensures that the groups being compared are statistically similar at the onset of the study, providing a foundation for valid evaluation. In studies, blinding plays a critical role in minimizing bias that could affect the ascertainment of endpoints. By keeping treatment allocations concealed, it prevents the influence of knowledge on the behavior of participants and researchers, establishing a more objective research environment. Pre-specification in study protocols is of utmost importance to address multiplicity concerns. By carefully defining primary interests and objectives from the outset, we substantially reduce the likelihood of obtaining statistically significant results purely by chance. Understanding biomarker positivity stands as a crucial element for determining drug efficacy in clinical trials. This understanding helps delineate which patients are most likely to benefit from specific treatments, aligning therapeutic approaches with patient needs. The FDA's guidelines regarding patient-focused drug development provide critical insights into selecting meaningful endpoints. These guidelines prioritize clinical relevance while fostering attempts to minimize bias and variability, steering research toward more impactful outcomes. The importance of achieving statistical significance in drug effect evaluations cannot be overstated. It's essential to note that a lack of statistical significance does not inherently indicate a drug's ineffectiveness. Context and comprehensive analysis matter greatly. When blinding is impracticable, no treatment control trials become necessary. In these instances, participants receive standard care while others benefit from additional interventions, thereby ensuring a fair and balanced comparison of outcomes. Unfortunately, data manipulation, often termed fudging, can occur when only significant results are presented. Establishing proper pre-specifications for study parameters is vital in fostering both transparency and accuracy in reporting findings. Publicly registering study protocols enhances accountability within the scientific community. This proactive practice empowers peers to verify study intentions and results, effectively mitigating potential biases that may arise throughout the research process.